Okay, couple quick things as we get going here. First off, I'm going to pass around the sign-in sheet. Okay, big thing for this week is actually we will be having our second exam on Wednesday. So exam number 2 is on Wednesday. And so here during normal class time. Again, that is going to cover the unit of material from motility up through enzymes and energy lecture set that we finished before spring break then. We did start some exam 3 content, which was catabolic reactions. For instance, we started talking about things like glycolysis. We're going to finish talking about it today. We also talked about things like fermentation. Remember, that is exam 3 content, not exam 2 content then. Exam 2 is going to be the same format as the first exam, a mixture of multiple choice questions, some matching questions, and a short essay question or 2 then on that particular exam. Is there any questions about the exam, or is there any questions about the content on the exam? Also, don't forget the pathogens of interest. I do have the pathogen of interest study guide posted onto Brightspace as well. Remember, there's things like the interactive notes are useful for prepping for the exams, but I also then do have a study guides and study materials folder on Brightspace that has things like each lecture's worth of some study objectives, and then the pathogen of interest study guide. It just has all the pathogen of interest that are on the exam and all the slides that were relative to them in one spot to help with prep with that portion of the exam. But do remember that portion of the exam is about 10% of the exam over. So you really do want to make sure you know the pathogen of interest well. Okay. So that is going to be on Wednesday. That is actually the only assignment for this week. Okay. So since it's an exam week, we do not have any Brightspace quiz on Wednesdays, just the exam on Wednesday. No Brightspace quiz, no interactive notes due this week. Okay, so that's typically what I do during the exam weeks. Another quick reminder, midterm grades got posted before spring break. Obviously, it does not include, it only includes the first exam because we haven't taken the second exam yet. Midterm grades are on My Mount Mercy, selfservice.mtmercy.edu. The midterm grades, again, I made this announcement before spring break, but they reflect your lecture and lab grade combined as of last week before spring break. That, again, is the way we calculate the grades in this class is both lecture and lab count towards 1 grade for the course. Your final grade in the class is 2/3 your lab percentage and 1/3 your, 2/3 your lecture percentage and 1/3 lab percentage. So again, if you ever want to figure out what your grade is at the class in any given moment, you take your percentage in lab plus 2 times your lecture percentage, and then you take that number divided by 3. You're just averaging them out. Okay, but your lecture percentage kind of double counts versus the lab percentage is just single counts. And you're taking the average then. Questions about that? A grade of C or higher, that is a 72%. A C is required in this class to count towards the nursing major. All right, so before I go on to content for today's lecture, any questions about, again, exam 2 content before I keep going through lecture and exam 3 content then? Yeah. So the math problems will be on the exam. There will probably be 1 or 2 on the exam for sure. Most likely, if I put a math problem on the exam, it might be one of the short answer, short essay questions. And you are going to have to show work, for instance, then. Yeah. I do like diagram style questions in general. It just is I might also ask you to interpret diagrams that we went through class as part of multiple choice questions then as well. So I don't know if you're gonna have to draw one out, but you could actually have to interpret some of the ones we've went through as well. So it could be a mixture of them. I'm gonna have some diagrams and then questions over them. Could be some draw diagrams, yeah, where they're relevant. Same as the first exam. I was fairly obvious in the study materials, the study guides, and the Learn Interactive Notes. I was going to put a gram negative versus gram positive diagram on the first exam. I asked you guys to draw it out multiple times on different materials. Okay, if you guys do have other questions, please feel free to go ahead and interrupt me. I'll try to save some time at the end of class today to go through that a little bit as well then. I do want to make sure I'm not rushing through content throughout the semester, so I'm probably going to, unless you guys have more questions, I'm going to start with going through more exam 3 content though. I don't like to rush. I already rushed too much. But that's actually that's a biology class in college. It's always fast. It's always a lot. That's the same way probably with anatomy then as well. So biology is known as one of the harder college classes because we just cover a lot of content pretty quickly. And that's in all the biology courses. Okay. All right. We left off talking about glycolysis. What's the overall function of glycolysis? What's kind of the purpose of glycolysis? Break down glucose, but why break down glucose? To generate energy intermediates for the cell. We talk a little bit about the glucose. What's the chemical formula for glucose? You guys should know that probably. C6H12O6. Glucose is a 6-carbon sugar, is basically what that is just saying here. This is a kind of organic chemistry diagram, by the way, if we're reading these. Every point is a carbon. 1, 2, 3, 4, 5, 6 carbons. So I'm using some of these shapes, some of these as a shorthand for certain types of chemicals. But I'm really going to focus a lot on the number of carbons involved in some of this. And so my little hexagon here is also going to be a 6-carbon glucose. It's just a really shorthand way to represent that. Glucose, C6H12O6, it typically is a ring-shaped sugar, hence also my ring shape up here. With that, though, it is pretty dang stable. It doesn't like to react. The cell has to spend a little bit of energy to get the process of glycolysis started. This is called the energy investment phase. It spends a ATP, adenosine triphosphate. That goes in. What comes out is a adenosine diphosphate. But again, this is triphosphate versus di. Tri means 3, di means what? 2. Diphosphate, then the third phosphate, this third phosphate ends up going on to glucose and making something called glucose 6-phosphate. So I'm just tracking the phosphate group. The phosphate group adds it negative charge to it, and it also just makes that glucose, that sugar, a little bit more reactive. Sometimes that glucose 6-phosphate can get converted into other sugars. So that, for instance, might be a 5-carbon ribose sugar over here. What was the name of the pathway that can convert sugars into other sugars? We talked about that before spring break. I know it's been a whole week. Hopefully you guys had some fun, not too much partying. I don't know. Anyways. So there is a pathway that converts sugars of one type into other kinds of sugars. Anybody remember the name of that pathway? PPP, which stands for pentose phosphate pathway. But again, this can convert glucose 6-phosphate or other sugars into other sugars, like, for instance, ribose sugar. And a ribose sugar is a 5-carbon sugar, for instance. But ribose, you use that to make things like nucleic acids, like DNA, deoxyribose. Okay, deoxyribonucleic acid, or ribonucleic acid, ribose nucleic acids. Okay, RNA. Glucose 6-phosphate can sometimes go over here, but it can also sometimes go instead into glycolysis. If it goes into glycolysis, you spend another ATP. And again, you get an adenosine diphosphate out, but then you take that third phosphate and you attach it onto that sugar. There's actually a couple different steps. One of the chemical steps is sped up by an enzyme called phosphofructokinase. Anytime you're looking at this, though, what does this ASE ending mean? ASE. Does anybody know what that is always going to be in reference to? Yeah, ASE just means you are dealing with an enzyme. Anything that ends in ASE ending is some kind of enzymes. And for instance, the name of the enzyme also typically gives you what is its function? And you read it backwards. But in essence, phosphofructokinase is an enzyme that adds a phosphate group to fructose sugar. So there's a couple chemical steps, but the glucose 6-phosphate gets converted into fructose sugar, which has then got another phosphate added in, and this is called fructose 1,6-bisphosphate, bis for 2 phosphates. It's called 1,6-bisphosphate because one of the phosphates is coming off of the first carbon in the sugar molecule. The other one's coming off of the 6th carbon in the sugar molecule, in essence. And so that is where that name comes from. But you guys might have heard of other enzymes, for instance, like DNA polymerase. DNA polymerase is an enzyme that polymerizes or makes DNA. So the names here are descriptive also of their function and can be a clue to kind of help you. They're not going to give you it necessarily without studying, but it gives you a hint to help you remember some of these enzyme functions. Now, if fructose 1,6-bisphosphate, you're spending another ATP. So you're spending one ATP to make glucose 6-phosphate. You're spending a second ATP to then end up making, through a couple chemical steps, fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate always goes into glycolysis. This is what is called, always goes into glycolysis. This is what is called the committed step. Once you make fructose 1,6-bisphosphate, it's always going to go into glycolysis, and only fate is to go into glycolysis. Again, glucose 6-phosphate can go up a couple different things, but fructose 1,6-bisphosphate is always going to go into glycolysis. I'm going to come back then to that chemical diagram we started going through. Again, glucose is a 6-carbon sugar. So we got sugar 1, a carbon, our 6-carbon sugar, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. Each little sphere in my little diagram is meant to be a shorthand for a carbon in glucose. Glucose goes into glycolysis, but they have to do that energy investment phase first. You spend an ATP here, you spend a second ATP here to prime that glucose and make it more reactive. This is then going to be the fructose 1,6-bisphosphate, okay, in terms of this one. Again, it's a 6-carbon sugar with 2 phosphate groups attached. I'm showing the same thing in a couple different ways. Once you form fructose 1,6-bisphosphate, it always goes into the remaining chemical reactions of then glycolysis. And typically, the very first thing is, it turns out this molecule, once you've made it, once it is fairly got these phosphates and groups, it actually means it's not very stable. So these phosphate additions are destabilizing or making the molecule more reactive. And almost immediately, it gets split into 2 3 carbon molecules, each with a phosphate attached. Again, my points are 1, carbon 2, carbon 3. Again, it splits into over here another carbon 1, carbon 2, carbon 3, so on. But you got these 3 carbon molecules that are then got a phosphate group, and these are called glyceraldehydes. So again, these glyceraldehydes are the same thing. These are the glyceraldehydes. They're the same thing, and that's meaning I'm just showing the same thing in a couple different ways. These are then kind of go through 2 different things, but these are the same chemical reactions here as here. The 2 glyceraldehydes that are formed, or sometimes they're referred to as glyceraldehyde 3-phosphates. One of them will go through this part, this other one will go through this part, but this is the same set of chemical reactions. They go through the same chemical reactions. I'm just showing the 2 different kind of wings, but it's the same thing happening to each of the glyceraldehydes formed. The first thing that happens here is that we have NADH being made. Now remember, NAD plus is the version of NADH that is not storing energy. You can convert it into NADH. This is the version that is storing energy for the cell. But to do that, you actually have to add on 2 electrons. Okay? So when 2 electrons, high energy electrons, come onto NAD+, it converts it into NADH. NADH is storing energy in the form of those high energy electrons. But it is a cycle. Those electrons can come off onto something else, say just molecule X. That will recycle the NADH back into NAD+. So electrons come on, you got NADH. Electrons come off, you got NAD+. This is the version that is actually the energy intermediate that is storing energy, though. What happens in this case is, where do those electrons come from? In this particular chemical reaction, you're stripping a hydrogen carrying 2 electrons off of this. So what you're doing is you strip a hydrogen carrying 2 electrons. You strip that off of what had originally been glucose, but is now glyceraldehyde. And those electrons go on to NAD+ to make it NADH. You get those electrons off by replacing them with an inorganic phosphate group. So this is an inorganic phosphate group coming in. When the phosphate group comes in onto the glyceraldehyde, it bumps a hydrogen and 2 electrons off. Those hydrogen and 2 electrons go into NAD+, converting it into NADH. And what you've done is you have made an energy intermediate. This is the first bit of energy that's getting harvested out of glycolysis then. And you make a NADH off of this one, glyceraldehyde, you make another NADH off of that other glyceraldehyde that was being formed. Questions about that so far? So at the end of this, you got a 3 carbon molecules, 2 phosphate groups attached, and you're making some NADH. And so these guys right here are like this guy right here, okay, at the end of this stuff. This is an example. These electron transfers are an example of what are called oxidation reduction reactions. Oxidation reduction reactions are any type of chemical reaction that involves electron transfer. And sometimes they're also referred to as then redox reactions as well. So oxidation reduction reactions, redox reactions, those are 2 different terms, 2 different names for the same thing. But they also always involve electron transfer. So you might have molecule A, an electron is going to get transferred from molecule A onto molecule B. And that electron transfer is then referred to as a redox reaction then. Electron transfers. Sometimes electron transfer is endergonic. It takes some energy. Sometimes electron transfers, though, are exergonic. It can give off energy over the course of that electron transfer. So for instance, when NADH loses electrons. When electrons come off of NADH, remember, when electrons come off to, say, some other molecule, molecule X, when electrons come off at NADH, that is going to be an example of a exergonic reaction. It gives off free energy for the cell to do work. Okay. It also recycles it back to NAD+, and if that can accept new electrons at some later time point, so that can get reused and recycled and regenerated. Questions about that so far? Oxidation is the process of losing an electron. So for instance, we're saying molecule A in my diagram is being oxidized because it is giving up or losing an electron. Reduction is the process of gaining an electron. And in this particular diagram, molecule B is being reduced or undergoing reduction because it is gaining an electron. In any redox reaction, there's always one thing that's losing an electron and something else that's gaining an electron. Anybody happen to know the acronym to help you remember this? The difference between oxidation and reduction? There's a mnemonic device for remembering this. Oil rig. And what it is that stands for oxidation is loss, reduction is gain. What's happening during glycolysis or during NADH? For instance, glycolysis, there's a bunch of high energy electrons on sugars like glucose. Those high energy electrons are a good source of energy for the cell. But during something like glycolysis, electrons come off of glucose or what had originally been glucose at the beginning of glycolysis. And those electrons go on to NAD+ to make NADH. Again, that NADH is then storing high energy electrons, and it's an energy intermediate or energy currency for the cell then. NADH stores that energy, but it can also then be used. NADH at some later time point can then donate those electrons to something else in the cell. So for instance, that something else can be something like NADH, and those electrons get transferred over to oxygen gas, for instance. And we're going to talk about that when we talk about respiration. That converts the oxygen gas into water. But that is going to be a very exergonic reaction. It gives off a lot of free energy the cell can use to do work then. So these electrons coming off of NADH onto something like oxygen, that's actually very exergonic. And we'll talk about that as we go through things a little bit more. But these are just examples of redox reactions. Glucose is glucose being oxidized or reduced here? Glucose is being oxidized here because you're taking electrons off of glucose. Glucose is losing electrons. The electrons are going on to NAD+. So is NAD+ being oxidized or reduced? When NAD+ gains the electrons, it's going to be reduced then. So the electrons are going from glucose to NAD+. Again, when the NAD+ accepts those electrons, it converts it into NADH. When glucose, over the course of glycolysis, loses electrons, it gradually gets converted into something called pyruvate, which we'll talk about more here in just a second. The other steps, there's some other chemical steps, but in essence, what happens in the rest of glycolysis then is you have this, what's called bisphosphoglycerate molecule right here. It's a 3-carbon, 2-phosphate molecule, okay? What ends up happening is through multiple chemical reactions here, the phosphates get stripped off. When that phosphate, this is again the phosphate right here, you have adenosine, you have adenosine diphosphate. This phosphate gets clipped off, and when you clip that off, you make then adenosine triphosphate. So you remove a phosphate here, add it onto adenosine diphosphate to make adenosine triphosphate. But you're doing that process here, 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 and here. So how many total ATPs am I making? 1, 2, 3, 4. So I'm making 4 ATPs here towards the end of glycolysis. This process of stripping the phosphates off of the sugars, these are sugars through here, 3 carbon sugars. The process of stripping those carbons off of sugars to then add them on and make adenosine triphosphate is then called substrate level phosphorylation. You're stripping phosphates off of a sugar substrate in this case to make ATP. Substrate, again, level phosphorylation is going to make ATP. How many net ATPs did we make then? Over the course of glycolysis from the start to the end, what's my net ATP production? Remember, this is spending an ATP up here. We're spending an ATP here. So we're spending 2, but we're making 4. So what's my net product? Or my net profit, sorry. 2 ATP. So if we overall look, we're getting 2 ATP in here, 2 ATP out, but those kind of cancel one another out. And then we're left with 2 extra net ATPs being produced in the end. What that means is if we're looking at the net gains from glycolysis, this is glucose goes in. So again, glucose is going into the pathway, into glycolysis pathway. Glucose comes in, but the net products from glycolysis then are 2 NADHs that's getting produced and 2 net ATPs that are being produced in terms of the energy intermediates. So these are the energy intermediates that are being made from glycolysis. The other thing we get out at the end of glycolysis is 2 pyruvates. So this is what you then get out. And I didn't go through it, but this little molecule at the end, this 3-carbon sugar alcohol at the end is called pyruvate. Sometimes it's also then referred to as pyruvic acid. So they are the same thing, though, really, honestly. Okay. So the overall function of glycolysis is to break down glucose to make energy intermediates. Glucose goes in to glycolysis. What you get out of glycolysis is 2 NADHs, 2 net ATPs, and 2 pyruvates. So again, this is what the glucose goes in. This is what you're getting out. That. Questions about that? Where in the cell does glycolysis take place? Yep, in the cytoplasm. The other thing to keep in mind is the things I want you guys to know with some of these metabolic pathways. I always want you guys to know what goes in. I want you guys to know where it takes place in the cell. I want you to know what comes out of these these metabolic pathways we're going to talk about. But where glycolysis is in the cytoplasm, glucose is going to go in, and what you get out is 2 NADH, 2 ATPs, and 2 pyruvates. Questions about that? All right, with glycolysis, there is a little bit of a problem that we also want to talk about. Glycolysis, this is a different way for me to just represent glycolysis. Glucose goes in, and then I got some pyruvates coming out. I got some NADHs coming out. I got some ATPs coming out. So this is a different way for me to just show the glycolysis pathway from start to finish with some of the intermediate steps. Each one of these little guys is supposed to be an enzyme overseeing a chemical reaction. For instance, this would be the phosphofructose enzyme, phosphofructokinase enzyme. This width of my orange line is going to be the amount of a particular chemical inside the cell. Typically in the cell, there's lots of glucose. The wider my line, the more there is of that particular chemical. So here, I have a pretty wide line. There's usually lots of glucose in the cell. There's usually lots of pyruvate in the cell, one of the end products of glycolysis. But all these intermediates rapidly go through the different steps of the metabolic pathway. Remember, a metabolic pathway is a series of interlinked chemical reactions. So all the intermediates, like for instance, fructose 1,6-bisphosphate, there's usually not very much of that in the cell. And it gets rapidly goes into the next step of glycolysis, which gets converted to the next intermediate, which then gets rapidly converted to the next intermediate, the next, until we get to the end of the pathway. Glycolysis usually happens very, very fast and very, very efficiently. But there's a problem with it. One of the things is glycolysis is sometimes too good at its job. Glycolysis will burn through the glucose in the cell, convert all that glucose in the cell really quickly. And it keeps stripping electrons off of the glucose and the things that glucose gets turned into. Just keeps stripping more and more and more electrons off of them very, very fast. As those electrons get stripped off of these sugars, they go onto NAD+, and those electrons convert it into NADH. And sometimes glycolysis does this so well that it converts almost all of the NAD+ in the cell into NADH. So it rapidly converts the NAD+ in the cell into NADH. And then you're left with no NAD+ left in the cell, or very, very, very little of it. In essence, the cell runs out of that NAD+. If that happens, NAD+ is a necessary reactant at this chemical step. So NAD+ is going in right here. But reaction runs out of NAD+, to use. And so what ends up happening, and this chemical reaction can't happen, but if this chemical reaction can't happen, this one can't either, and this one can't either, and anything downstream doesn't happen. And so when the cell runs out of NAD+, it stops the process of glycolysis. That is bad because are you making energy for the cell anymore? Is the cell getting any energy intermediates? Any NADH being made? Any ATP being made? No. And the cell locks up. So the cell's not going to be getting energy. It's actually worse than that. Looking at these dynamic cell, like NAD+, NADH, pyruvates, glucose is one of those sugar intermediates. All of these are floating around the cell. We like to think of them as nice and neat and ordered. So now I just took it and I put them next to one another, but we can see how much glucose is in the cell, or how much NADH, or how much pyruvate. But they're all jumbled. See things like chemical concentrations. Glycolysis is functioning like it's supposed to, but all those intermediates are at low concentration. However, again, my size of my font is chemical concentration. So I have a little tiny font of NAD+ here. That means the cell is running low on NAD+. If you don't have NAD+, this chemical reaction locks up. And you would go from very, very little fructose 1,6-bisphosphate in the cell to tons of fructose 1,6-bisphosphate in the cell. Tons and tons of it build up in the cell. which is not the way it normally is. Does that kind of make sense, everybody? Or questions about that? Okay. Now, you're not making NADH, you're not making ATP. That's bad for the cell. It's actually worse because when this builds up, when this chemical builds up to high level of concentration, it starts to interact with other enzymes it normally would never interact with. So when this particular chemical builds up to high levels of the concentration, it starts interacting with different enzymes. My green things here are different enzymes, and actually converts it into something called methyl glyoxal, which you guys might know better as antifreeze. Do you go drinking antifreeze? No. And it is actually toxic to the cells. So when glycolysis locks up due to a lack of NAD+, it makes toxic chemicals that can kill the cell if those chemicals build up then. So not only is it not processing energy, it makes toxic byproducts. Questions about that? So the cell needs a way to solve this problem. One of the ways to solve the problem is let's use up NADH in some anabolic reactions. Anabolic reactions will use up some NADH, but not fast enough. They don't use up the NADH in anabolic reactions quick enough, so the cell needs other solutions. One in the solution is typically glycolysis is always either coupled to fermentation or it's coupled to respiration. So glycolysis is done in most cells. Glycolysis happens in our cells, most bacterial cells. Not all. There are some species of bacteria that don't use glycolysis. But we're not going to talk about those too much because they're not important for disease. With the glycolysis, though, any organism that has glycolysis couples glycolysis to either fermentation or respiration reactions. In either case, one of the functions of either fermentation or respiration is to use up NADH. But again, if you use up NADH, you take electrons off of it, but that recycles it back to NAD+. Okay? So when you use up NADH, it recycles it back to NAD+ when those two electrons come off. Those two electrons are going to go into either fermentation or respiration. And the function of both, one of the functions of both, is to then regenerate the NAD+ chemical pools and keep glycolysis running. But they do it in different ways. Fermentation. Fermentation is one strategy to use up excess NADH and again, recycle it back to NAD+. In yellow is glycolysis. So my yellowish orangish, this is representing glycolysis on this side. Okay. With glycolysis, you're making things like 2-ATP, 2-pyruvates, 2-NADH. So those are glucose going in. They go through glycolysis. You get, again, 2-ATP, 2-NADH, 2-pyruvates. Fermentation is the part in red then. So fermentation is this one, okay, in red. The process of fermentation is it takes high energy electrons from NADH and puts them onto pyruvate. So you take the electrons from NADH, the two electrons from NADH, and they go onto pyruvate. This converts pyruvate into some type of fermentation product like lactate. What it also does, though, is as those electrons come off of NADH, it regenerates NAD+. NAD+ will feed back into glycolysis and keep it running. So fermentation, high energy electrons off of NADH are placed onto pyruvate. That regenerates NAD+ and keeps glycolysis running. But fermentation itself makes nothing else for the cell. The only thing that fermentation does, fermentation itself makes no energy for the cell. In fact, it probably wastes cellular energy. The only purpose of fermentation is to use up excess NADH, regenerate it to NAD+, and then that NAD+ funnels into glycolysis, and it's the glycolysis that is then making the energy for the cell. In fact, fermentation is probably wasting that NADH energy intermediate. And also, but not as toxic as methyl glyoxal. Question about that before I kind of keep on going with fermentation here a little bit. Okay. Turns out there's also many different kinds of fermentation. Different species of bacteria do different kinds of fermentation with different fermentation products. Fermentation always starts with NADH and pyruvates or pyruvic acid. Okay, so what goes in to fermentation is, again, the 2 NADHs plus the 2 pyruvates. What comes out then depends upon the bacterial species. So for instance, certain species of bacteria like Streptococcus, Lactobacillus, Bacillus, certain species like us, for instance, we can do fermentation sometimes as well. They have fermentation that produces lactic acid as the product of fermentation. So they do one type of fermentation that does lactic acid as a product, end product. Things like Saccharomyces cerevisiae, baker's yeast does a type of fermentation that makes ethanol and CO2 gas at the end of it. Propionobacterium produces propiononic acid, acetic acid, CO2. Clostridium makes these fermentation products. Escherichia coli, Salmonella makes these fermentation products. Enterobacter species make these fermentation products. Different species of bacteria do different kinds of fermentation and different fermentation products. But it always starts off with the NADH and the pyruvates going in. That's what starts the process of fermentation every single time. But the end products depend upon what bacteria species it is. Questions about that? All right, so different bacteria do different kinds of fermentation. This is just showing different fermentation reactions, but this might be the type of fermentation in one species. This might be a type of fermentation in other species. These are some types of species do these fermentation reactions. So like 1, 2, 3, 4, 5, 6. It's just showing different kinds of fermentations in different kinds of species. But again, it always starts with pyruvate and also the NADH. It always starts with those two things. Okay? That's what you start fermentation, but different species do different kinds of fermentation. And no, you don't have to have these memorized. That being said, also different bacteria fermentation products, many of them are commercially important. Ethanol, acetate, propionate, isopropanol, butanol. These are some common fermentation products that are used very, very commonly. If you guys have ever wondered where a lot of your vinegar comes from that you might buy from the store, it's bacterial fermentation. They grow giant vats of bacteria, and then they harvest the vinegar, the acetic acid, that's produced as a waste product at the end of fermentation. Ethanol, drinking alcohol, is basically from yeast. They grow baker's yeast in giant vats, they feed it sugar, and they get ethanol out. That they then, beer is just growing yeast, for instance, until it ferments sugars and makes the alcohol. Many of these fermentation products, though, are toxic if they build up to high level. So if there's lots of these fermentation products and they're being built up, it's going to be toxic to the cells that are producing. We use those fermentation products, and you guys are going to do this in lab if you haven't started already, to sometimes do diagnostic tests. We can actually use fermentation products to figure out what kind of bacteria are causing what kind of disease. For instance, this is a type of biochemical test called a phenol red test. Sometimes it's also referred to as a carbohydrate fermentation test. You guys are going to do this in lab. You guys haven't done this yet, right? No. You're going to do this in lab later in the semester. Phenol red or carbohydrate fermentation test, you can actually grow this. The media is going to start off red. So this is my uninoculated negative control too. So the media starts off red. If it stays red, and this was one where it stayed red, this is a negative for fermentation. this case, they fed it a sugar, they fed it glucose sugar, and they found out that Staphylococcus epidermidis, for instance, was negative for this fermentation of the glucose sugar. Stayed red. However, if it turns yellow, and here we see yellow for Staphylococcus aureus or for Escherichia coli, what that means is they're breaking that sugar down via a combination of glycolysis and fermentation. Most fermentation products are acidic. What ends up happening is there is a pH indicator in here called phenol red. Under neutral pHs, it's red. So neutral, that's negative for fermentation. Most fermentation products, though, are acidic. And so as they make these, if they do ferment the sugar and they make the fermentation products, it makes the pH, the medium, more acidic or lowers the pH. And as you get the pH lower and lower, that pH indicator changes from red to yellow. So these are positive results for it. You guys will also have a little upside down tube called a Durham tube in there. You don't have to worry about this too much. But you guys see how there's a little glass tube in here? It's hard to see, but there is a air bubble here. And it's actually a CO2 air bubble is what it is. That's also positive for fermentation, but more specifically, makes CO2 gas as one of the products of fermentation. So here you see the air bubble. That is going to mean that Escherichia coli does ferment the sugar, but also specifically makes CO2 gas as one of the fermentation products. Here you're seeing no air bubble in the tube, and so that does do fermentation, still a positive result, but without CO2 production. If you want, this is commonly done to do disease diagnostics, by the way. Fermentation capabilities. You take a culture from a patient, you grow it up for a day in this media, and then you can say, oh, this isn't doing fermentation, and based on the combination of symptoms of fermentation, it's probably this bacteria causing this disease. But if you take a culture from a patient and it does do fermentation, maybe it's a different species of bacteria, and that often can impact treatment, for instance, then as well. Questions about that? So again, sorry, I had this on the next slide. I kind of went through it, but this is a different way, just the same thing I just went through. Most fermentation products are acidic. So under neutral conditions, the pH indicator is red, but under acidic conditions, it turns yellow, and that's a positive result for fermentation. You also got that little upside down Durham tube. And if you have any CO2 production, that's also a positive for fermentation, but even more specifically for CO2 gas as a product of the fermentation. There are other tests that test for fermentation, and you guys might do some of these in lab then as well. So there, for instance, there's something called the MRVP test. This is actually two different tests, a methyl red test and a Voges-Prosker test. Methyl red tests for stable fermentation products. Most fermentation products are unstable and they break down after a few days. There are some fermentation products that instead are much more stable after about 5 days. And so the methyl red test, you add a pH indicator after the culture's been growing for about 5 days or so. And if it turns color, then it is a positive result for those stable fermentation products. However, methyl red is actually yellow under neutral conditions and then red under acidic conditions. It's a different pH indicator. So for the methyl red test, it's actually red is positive instead of yellow. Voges-Prosker test for specifically is butanediol produced as a result of fermentation. So it looks specifically for this as a fermentation product. Some species can make that, most species don't make that, but there's a few important pathogenic species that make butanediol as a result of their fermentation. And so this one in the Voges-Prosker test, you actually can look for that product and see if it's present or not. Fermentation we can use fermentative capabilities to diagnose disease. That's really what we're doing often with the phenol red test or the Voges-Prosker or the methyl red test. We're trying to use the fermentation capabilities. Can they do fermentation or not? What fermentation products are produced? We can use that to diagnose disease, but fermentation causes other stuff. For instance, pipes, metals, wiring in houses, hulls of ships, metals. Fermentations make acids. Acids over time can etch and break down metals and cause them to degrade over time. So in many cases, fermentation species can corrode things over time. Also, fermentation is one of the reasons we get cavities or caries. So bacteria stick to teeth. They especially like to get in the gum pocket, the area between your gums and your tooth. They really like to grow up there. Anybody have a nice sugary breakfast and their teeth start to feel fuzzy? That's bacteria growing up in your mouth. They like to eat the sugars. They like to do fermentation. As they do fermentation, they make acids. The acids will then etch and eat away at the enamel of your teeth, creating holes. That can then lead to infections of the tooth that are cavities or caries. So you get cavities or caries because of the bacteria growing in your mouth doing fermentation. Okay, questions about that? All right, I'm out of time for today. We're going to pick up on, well, Wednesday we have the exam. Friday, we're going to have a pathogen of interest, but we're going to talk about glycolysis coupled to respiration then instead.